Let's talk about action potential. So here we have our neuron, and I'm going to explain action potential and synaptic transmission. In order to talk about action potential, we need to talk about and explain resting potential, which is also known as ready potential, because we are ready to send an impulse. So basically all you need to know here is that we have the outside of the neuron, we have the membrane of the neuron, and we have the inside. The outside has the chlorine, sodium, and calcium ions, and that makes the outside positively charged. And then we have the inside of the neuron with our potassium ions, and that makes the inside negatively charged. In addition to the potassium, we also have our large negative ions, as you can see in green. And the combination of these factors makes the membrane charged at negative 70 millivolts. Additionally, the sodium-potassium pump, as you can see in the middle in orange, pumps 2 potassium out and 3 sodium in. Also, a little bit of potassium does leak out into the outside because it has a greater permeability. All right, let's get into action potential now that we know the basics. So first and foremost, we need a stimulus to trigger an action potential. This triggers what's called depolarization, which means, and this is called getting closer to threshold, and so we get to negative 55 millivolts. At negative 55, the sodium doors open, and the sodium ions rush in, and this reverse polarizes our neuron to positive 30. Basically what that means, reverse, our charges are switched. So now the outside is negative and the inside is positive. So at positive 30, the sodium doors close because they're all done sending stuff in, and the potassium doors open, so the potassium rushes outside. And so now this is called hyperpolarization because so much potassium goes out of the doors and is leaking out that the charges switch again, and now it's at negative 90. And the potassium doors close. So basically, the potassium and the sodium have basically switched spots. Then we have what is called the refractory period, where our sodium-potassium pump returns the ions to their original position. It's basically resetting the neuron back to resting potential after you send the action potential. All right, so what's the significance? Like, why do we need action potentials? Well, one action potential triggers another action potential, and another, and another, and another, and so they continue down the axon, and this is called an impulse. But what happens when we reach the end, which you can see circled? This is where we send the impulse and neurotransmitters to an effector across a synapse. The effector is basically anything receiving the impulse, so that could be another neuron, a muscle, or a gland. So how does synaptic transmission work? So we have our receptors on the dendrites over here, and then we have neurotransmitters that are being sent across this gap. So it reaches the end of the neuron, and it must be cast across the synapse, as I mentioned, this gap, and it moves from the presynaptic neuron to the effector. If it's another neuron, you can also call it the postsynaptic neuron. All right, so depolarization. We have an action potential. This causes depolarization to negative 55. So this causes the calcium and the sodium doors to open, and these ions flow inside. These positive ions reverse polarize to positive 30, and the synaptic vesicle, which is negatively charged on the outside, migrates towards the cell membrane. So the synaptic vesicle is attracted to the now positively charged membrane. So previously they were both negative were both negatively charged, and so they would repel each other. but now that the membrane is positively charged, the synaptic vesicle is attracted to it, and it moves towards the end. And here we have our labels. So we have our calcium ions, we have our neurotransmitters inside the synaptic vesicles. Vesicles are kind of just like bags. Then we have the neurotransmitter receptor on the postsynaptic neuron. And then the synapse is this big gap. So the neurotransmitters are kind of spit out, essentially. They're released by exocytosis across the synapse. And some, not all, it's kind of just like spit out in random directions. Some bind, some others, some don't. So some bind to receptors, as you can see with the arrow. So what happens to other neurotransmitters that don't bind to receptors? Neurotransmitters that don't bind to the receptors on the postsynaptic neuron, gland, muscle are either eaten up by enzymes or sucked back up into the presynaptic neuron, where they'll be repackaged and reused. This is called reuptake. So it's kind of like recycling. So, you spit out all these neurotransmitters, only some bind to the receptors, and so you suck the unused ones back up and you recycle. And additionally, the enzymes eat up the neurotransmitters. They kind of tear them apart, and so these little shreds are also sucked back up and repackaged. All right, so there are two types of neurotransmitters. We have excitatory and inhibitory. So we have excitatory that causes depolarization, and it moves the neuron closer to threshold, and it's more likely to have an action which means that you're more likely to have an action potential. It opens the calcium and sodium doors. The inhibitory, on the other hand, they're opposites, causes hyperpolarization and moves the neuron further from threshold, which means it's less likely to have an action potential. It opens the chlorine and the potassium doors. All right, let's talk about excitatory first. Excitatory neurotransmitters are basically a stimulus in the way that they depolarize the membrane to -55. So the calcium or sodium or both open at -55, and these ions rush in towards the neuron. And so basically, these ions neutralize the inside of the neuron, and the charge moves closer to zero, aka threshold, because the difference between the two sides decreases. And this makes it much more likely for a neuron to have an action potential and therefore send an impulse. So basically, the inside was negative, but then all these sodium and potassium ions rushed in, and they made the inside super positive. And so therefore, it kind of neutralized neutralized the inside. All right, inhibitory neurotransmitters, on the other hand, are the opposite, as we talked about. And they hyperpolarize the neuron, -90. And this opens either the chlorine or the potassium doors. Those flow inside, or the chlorine flows inside and the potassium out. So as I said, K+ moves out, chlorine moves in. This essentially makes the inside super negative and the outside super positive. And this increases the difference between the sides' charges. The charge is now far from threshold, and causing an action potential / sending an impulse will be very difficult. Thank you for listening, and I hope you liked this presentation.